everyone, this video is about B cell generation, activation, differentiation and humoral immune response, and this particular study note is developed by EPAG Pathshala team, Government of India, MHRD. So I would like to express my sincere gratitude to the development team for developing this study note for immunology. Thank you so much. Now let's have a quick introduction about this. B lymphocytes were discovered by Bruce Glick, Ohio State University in 1954 in the bursa fabricis, a lymphoid organ in the cloacal region of chicken. Their origin was traced down to the evolution of adaptive immunity in jawed vertebrates about 500 million years ago. Lymphocytes are the cells which express diverse antigen receptors that recognize a wide variety of foreign substances. This diversity is generated during the development of mature B and T lymphocytes from precursor cells that are naive and do not have antigenic receptors, hence do not recognize any antigens. The principal events that occur during the production of plasma B cell and memory cell can be categorized into three stages: development of B lymphocytes, activation of mature B cells, and differentiation of activated B cells into plasma and memory cells. The process by which these bone marrow derived pluripotent cells are converted to mature lymphocytes are called lymphocyte maturation. Miller and J.F. Michael, 1968, demonstrated antibody producing B cell arose from bone marrow. The antigenic response leads to the primary humoral immune response with the production of IgM followed by secondary humoral immune response of IgG. B lymphocyte comprise 5% to 15% of total cell lymphocyte population, form and mature in bone marrow. Secrete specific immunoglobulins in response to antigenic stimulus, hence antibody mediated immunity. Then express unique surface immunoglobulin ISIG, mainly of IgM or IgD types, wherein mucosal associated lymphoid cells express IgA class immunoglobulin. The other markers on mature B cells are CD16, an Fc receptor for immunoglobulin, CD11 and CD35. They also express complement receptors and MHC2 molecules. Two subtypes of B lymphocytes are T cell independent cells, do not require help of Th cells for the production of immunoglobulins, recognize monomeric sugar or lipid antigens of microbes. Then T cell dependent cells require Th cells for production of T cell dependent cells require Th cells for production of immunoglobulins. The scanning electron micrograph shows B lymphocyte. T and B cells are distinguishable by light microscopy but can be differentiated experimentally, indistinguishable by light microscopy, and can be differentiated experimentally by probing their surface receptors. B cell development. Mammalian B cell development encompasses a continuum of stages that begin in primary lymphoid organ, human fetal liver, adult bone marrow, with subsequent maturation in secondary lymphoid tissues like lymph nodes and spleen. The functional endpoint is terminally differentiated plasma cells. The maturation of lymphocytes, both the B and T, follows sequential events like early maturation involving commitment and proliferation, expression of antigen receptor genes, and selection of mature receptors. Before birth, the B cells are usually generated in the fetal yolk sac, fetal liver, and to some extent in the fetal bone marrow. In the hematopoietic cells of the bone marrow, commitment towards B lymphoid lineage cells depend on the expression of transcription factor PAX5. Development of B cells. Early maturation and growth factor mediated expansion, antigen receptor expression, selection of repertoire, acquisition of functional competence, performance of effector functions, generative organs, peripheral lymphoid organs, self-antigen, foreign antigen. The early B cell development is characterized by the ordered rearrangement of IgH and L chains loci, and the Ig proteins themselves play an active role in regulating T cell development. The surrogate L chains is a heterodimer consisting of two proteins, alpha 5 and V pre-B. They pair with mu H to form the pre-BCR in murine and human pre-B cells. B cells arise from the pro-B cells that have neither pre-BCR nor surface Ig. BCR expression is the prerequisite for B cell development and survival in the periphery. Pre-B cells, the receptors mu heavy chain and lambda 5 pre-B are expressed in low levels on the cell surface in association with Ig alpha and Ig beta. These immature receptors transduce signals for further migration and also rescue the developing lymphocytes from apoptosis. Hence, this implies the first checkpoint in B cell maturation to ensure that the somatic recombination is successful. The pre-B cell undergoes proliferation to give rise to immature B lymphocytes. In this stage, the developing B cell produces either kappa or lambda light chains due to allelic exclusion and finally combines with mu chain of IgM. The next checkpoint happens when the mature B cells with complete receptor heavy and light Ig signal for continued survival, proliferation and maturation. Pre-B cell receptor. Light binding site, surrogate light chains, disulfide linkage mu heavy chain, mu lambda 5, pre-B, IgB, Ig beta, Ig alpha, IgM signal transducing. Antigen binding site, disulfide linkage, mu heavy chain, lambda or K light chain, Ig beta, Ig alpha, IgM signal transducing. So early maturation of B lymphocytes. Pluripotent stem cells of bone marrow give rise to B lymphocyte progenitor cells or pro-B cells. Pro-B cells express mu heavy chain on its surface. Functional antigen receptor genes are produced in pro-B lymphocytes by the process named as somatic recombination, in which a set of inherited germline DNA sequences are separated and recombined by enzymatic deletion and relegation. Terminal deoxyribonucleotidal transferase (TDT). This diversity of the lymphocyte is generated during the joining of gene segments and is not dependent on the presence of antigens. Antigen-induced B cell activation and differentiation in secondary lymphoid tissues are mediated by gene expression that give rise to germinal centers or GC. The GC reaction is characterized by clonal expansion, class switch recombination at IGH locus, somatic hypermutation, SHM of VH genes, and selection of increased affinity for a BCR for its unique epitope through affinity maturation. The somatic recombination SHM is carried out by half RAG recombination activating genes. Both SHM and class switch recombination are mediated by an enzyme called activation induced steady and deaminase AID. AID produce antibody diversification either by receptor editing or partitioning more directly at the DNA level. Participating. Okay. So this is the B cell development in detail. We will be reading the description below. Broad outline of B cell development stages in mice and humans. BLA, BLB and B10 populations are less well characterized in humans. CLP, common lymphoid progenitor. SHM, somatic hypermutation. CSR, class switch recombination. B1A, natural AB, B1B, adaptive AB, B10, IL-10. Short-lived plasma membrane. CLP, pro-B, large pre-B, small pre-B, fetal liver and bone marrow. Immature T1 and T2, marginal zone B spleen. Follicular B cell, activated clonal expansion, short-lived plasma cell, memory cell, plasmablast, short-lived plasma cell, then germinal center, apoptosis, clonal expansion, plasmablast, plasma cell. Marrow stromal cell derived IL-7 promotes V to DJ rearrangement and transits survival proliferation signals. At least 10 distinct transcription factors regulate early B cell development, namely E2A, EBF, and PAX5, multifunctional transcription factor for B cells. PAX5 regulates expression of at least 170 genes, a significant number of them for B cell signaling, adhesion, and migration to mature B cells. Pro-B cells have ALA4, which helps them to directly bind to stromal cells having VCAM1. Pro-B cells have receptors for tyrosine kinase c-KIT, which gets activated by the stem cell factor of stromal cell. This activation converts pro-B cells to pre-B cells. Early maturation of the pro-B cells also happen due to stimulation by IL-7 produced from the stromal cells of bone marrow, leading to high toxic mitotic activity and marked increase in cell numbers. Transcription factors Ikaros, E2A, PAX-A2, SIGM, SC probe early pre-B cytokines, SCF, IL-7, PAX5, T, NK, DC, neutrophil, M, beta. Selection process that form mature B lymphocyte repertoires. Pre-B or immature B cells are positively selected due to recognition by self-antigens and further ensured that all B cells that mature should express functional antigen receptors. The B cells also encounter negative selection during the short period after antigen receptors are expressed. The negative selection of developing lymphocytes is an important mechanism to maintain tolerance towards the self-antigens. This process is explained by Namazi and Burt, 1990, and follow the steps and encountering self-antigen. The development of immature B cell is arrested. Self-antigens that mediate negative selection are polyvalent and deliver strong signals to IgM expressing immature B lymphocytes. To protect themselves from negative selection, B cells undergo the process of receptor editing, thereby gene rearrangement of secondary receptor antigens allow B cell to replace inappropriate receptor with a new receptor. The edited light chain, usually kappa light chain, with the original heavy chain in them expressed. These IgM are not self-reactive mature to the next level. Diagram explaining the process of receptor editing. Immature B cell, receptor editing failed, light chain and changed. Self-antigen, receptor editing successful, light chain changed. Self-antigen can bind. Apoptosis of B cell, no binding of self-antigen. B cell survives. As attainment of functional competence by B lymphocytes. In the last phases of B cell maturation, the lymphocytes acquire the ability to respond to antigens and generate the effector mechanisms that serve to eliminate antigens. Expressions of a variety of cell surface markers occur on B cell along with normal levels of IgM and IgD. These cells enter the lymphoid follicles, can survive for a few weeks until they encounter their specific antigen or die. The final stage of development of B cells are the plasma cells, which have categorized the antigen and been stimulated by T cell derived cytokines. These plasma cells reside in spleen and lymph nodes found in the cytoplasm and secrete the antibodies such as IgA, IgM, and IgG. This change in production of surface IgM to other antibodies is known as class switch. Plasma cells are large lymphocytes with a large cytoplasm to nucleus ratio with basophilic cytoplasm and nucleus with heterochromatin, a characteristic cartwheel-like appearance. It has an extensive Golgi and abundant rough endoplasmic reticulum suited to produce immunoglobulin. The lifespan of plasma cells is of 2 to 3 days and they do not divide. The term plasma cells was coined by Waldever in 1875. Memory B lymphocyte. The cells are functionally and physically different from new B cells and stay longer. They have membrane IgG, IgA or IgE and high levels of ICAM1 and CR. Low levels of antigen do remain in the system to maintain the memory cell population. Then B cell activation. In response to foreign antigens, the proliferation and differentiation of B cell happen in the peripheral lymphoid organs. It's an antigen-driven process which results in the generation of antibody-producing plasma cells and memory cells are referred to as activation of B lymphocytes. In the absence of antigen, the new B cells die while memory cells can last for weeks or even months. The activation of B cells follows a distinct sequence. The recognition phase. Foreign antigens interact with IgM or IgD expressed on mature B cells. The proliferation phase, clonal expansion of antigen-stimulated B cells. The differentiation phase, generation of antibody-producing plasma cells and memory cells. Recognition phase, proliferation phase, clonal expansion, then the differentiation phase. Antigens bind to the membrane immunoglobulin molecules of mature B cells. Antigens bind to the membrane immunoglobulin MIG molecules of mature B cells. Antigen binding to the B cell receptor complex induces the clustering of membrane receptors that initiate the process of activation. The bound antigen is then endocytosed by B cells hydrolyzed, processed, and presented on the surface of the B cell. BCR is comprised of antigen binding MIG and signal transducing dimer of the Ig alpha and Ig beta heterodimer. The cytoplasmic domains of Ig alpha and Ig beta heterodimer have a tyrosine rich motif ITAMs immunoreceptor tyrosine based activation motif. Signaling through B cell receptor complex, tyrosine in the ITAM is phosphorylated by the tyrosine kinase like ILN, BLK, and FIN. Further, autophosphorylation of other tyrosine kinase like SYK finally culminates into a series of signaling cascades. Transcriptions of cytokines and immunoglobulin genes happen due to these signals, which ultimately lead to the activation of B lymphocytes. Depending on the nature of the antigen, B cell activation either require direct stimulation by TH, the thymus-dependent antigen, or they are activated without TH, the thymus-independent antigen, and thymus-dependent antigens involve contact-mediated activation of B cells and C and cytokine-mediated activation of B cells. The chance encounter of the antigen with its specific B cell and T cell depends on the continuous recirculation of lymphocytes into the peripheral lymphoid organs. The fundamental feature of a T-dependent B cell activation is the interactions that occur between antigen-stimulated specific B cell and T cell. This linked T cell interaction is called cognate interaction for that particular antigen. Antigens such as lipopolysaccharides act as Th1 antigens and usually generate a weak immune response and the IgM type of antibody production. No memory cells are formed by T1 antigens and there is no affinity maturation by B cells. The T1 antigen is a polyclonal B cell activated polysaccharides and glycolipid that generate multiple repeating identical epitopes are considered as Th2 antigens. They are not truly T cell dependent as B cells exposed to Th2 antigen require cytokine derived from Th cells. Hence, they activate mature B cells and inactivate immature B cells. So, polyclonal activator B cell antigen with repetitious antigenic determinant, clustering of membrane Ig, cytokine mediated T cell help needed, antigen with multiple dissimilar epitopes, contact mediated T cell help. Then B cell differentiation. B cell differentiation into plasma cells, antibody synthesis and secretion depending on the action of various cytokines as well as interaction of CD40 and CD40L on B cells. The differentiation of mature B cells into antibody secreting plasma cells require the change in the processing of RNA transcription factors in such a way that transition must happen from its membrane-bound form to the secretory form with the processing of its heavy chain mRNA. The CD40 and CD40L interaction is also very important in class switching. Prior to binding of an antigen with the B cell, the antigen is engulfed by receptor-mediated endocytosis and processed into peptides. Antigen binding also triggers the upregulation of a number of cell surface molecules, including the MHC2 molecules and the co-stimulatory ligand B cell. Enhanced expression of these molecules highlights the potential of B cell to function as an antigen processing cell in T helper cell activation. At high antigen concentration, dendritic cell and macrophages can also act as effective antigen presenting cells for Th cell activation. The processed antigenic peptide in association with the class II MHC molecule is identified by the Th cell, leading to the formation of a T cell B cell conjugate. This results in directional release of Th cell cytokines and activation of CD40 on B cells. Although conjugation of a B cell with membrane proteins from activated Th cells allows its proliferation, differentiation results from both the membrane contact signal and cytokine signals. Cytokines secreted by the T cell help the B cell to multiply and mature into antibody-producing plasma cells. Cytokines perform two main roles in antibody responses: antibody amplification by B cell proliferation and differentiation, determination of the type of antibody produced by promoting isotype switch. B cell differentiation into memory cells. Repeated antigenic exposure increase the population of memory cells. Memory cells bear high affinity immunoglobulin molecules on their surface. These memory cells express BCL2 gene, a proto-oncogene whose gene products avoid apoptosis and hence they have long life. Next, humoral immune response. The term humoral immunity refers to substances found in the humors or body fluids. Humoral immunity, or the antibody-mediated immune system, is that offshoot of immune system which is mediated by macromolecules found in extracellular fluids, in contrast to the cell-mediated immunity. The physiological functions of humoral immunity is defense against extracellular microbes and microbial exotoxins. So, humoral immunity can be transferred from one individual to the other passively by transfer of serum antibodies. The humoral and cell-mediated branches of immune system work in an independent manner. In the humoral response, B cells bind to invading antigen, differentiating into antibody-secreting plasma cells. The antibody then interact with the antigen and sequentially resulting in its clearance from the body. In the cell-mediated response, the different classes of T cell recognize antigen presented on cells and subsequently produce cytokines. So, cytotoxic T cells mediate killing of altered cell type, cells. Steps in the development of humoral response. Proliferation phase. Humoral immunity involves the activation of specific new B cells to proliferate and secrete large amount of specific antibodies on first exposure to an antigen, either by infection or vaccination. B cell is the key player that recognizes antigens or pathogens circulating in the body fluids such as blood and lymph. Activation of B cells by soluble protein antigens mostly involves co-stimulation by T helper cells. Then effector phase. In the next phase called the effector phase, activated Th cells trigger specific B cells to proliferate and release antibodies. These antibodies are highly antigen specific. They bind to the invader and fight the infection. The B cells undergo a process of clonal expansion and differentiation, subsequently maturing into antibody secreting plasma cell which secrete antibodies. The plasma cell produce antibodies with antigen specificity identical to the antigen receptors on the activated B cells. Antibodies, the effector products of humoral immunity, are released and circulate through the body binding to antigens. Finally, as this response declines, a pool of memory cells remains behind. If the body is re-exposed to the antigen, these memory cells will recognize the antigen and respond much more quickly and effectively. Antigen recognition. B cells with complementary receptors recognize and bind to receptors. Antigen presentation. B cell internalize antigens and display it as processed epitope. Helper T cells binds to B cells and secrete antibody. Clonal selection. Interleukins stimulate B cells to divide repeatedly and form clones. Differentiation. Most clones differentiate into plasma cells and some become memory cells, rendering antigen harmless. Antibodies. The key players in humoral response. Antibodies are glycoproteins comprising of polypeptide chains, light and heavy chains, with attached carbohydrate. There are 5 classes of antibodies found in the humans: IgA, IgG, IgM, IgE, and IgD. These antibody classes differ in their heavy chain regions. The light chain have an important role in elicitation of immune response as they form a part of the antigen binding site. Among these, only IgM and IgD can function as the antigen receptor for naive B cells. Antibodies can be secreted, circulate, and bind to invading pathogens, sometimes labeling it for identification by other forms of the immune response. IgA present in gastrointestinal, respiratory, and urogenital tract mucosa, also present in saliva, tears, and milk, involved in colonization of pathogen. IgD, membrane immunoglobulin. It is part of membrane receptor of B lymphocyte. IgE, involved in allergic and parasitic response, interacts with basophils and mastocytes and associated with the histamine release. Then IgG, main immunoglobulin of acquired immunity, inherent property to cross placental barrier. IgM, monomeric or pentameric. It is a component of membrane receptor of new B lymphocytes, PCR, found in serum, secreted early in acquired immune response. Antibody provides protection to an individual in 3 main ways. Neutralization. They bind to the invading pathogens such as they inhibit their toxic effects and in neutralizing their effects. Opsonization. By coating the surface of pathogens, antibodies can enable accessory cells that recognize the Fc portion of antibodies to internalize and kill the pathogen by enhancing phagocytosis, neutralization, opsonization, and activation of the complement system. Complement protein can strongly enhance opsonization and can directly kill some bacterial cells. Other complement proteins can recruit phagocytic cells to cell of infection. Primary and secondary immune response. The secondary response of both B2 T cells is observed following subsequent encounter with the same antigen and is more rapid, leading to the activation of previously generated memory cell. This has some quantitative and qualitative difference from the primary response. The drastic difference between the first and second exposure to the antigen reflected by the antibody titer in the serum is quite evident. The memory B cell repertoire generated helps intensify this response. The graph below depicts the primary and secondary humoral immune response. B cell functions in addition to antibody production. In addition to their essential role in humoral immunity, B cells also regulate or mediate many other functions essential for immune homeostasis. The B cells are required for the initiation of T cell immune response. This is carried out by antigen-specific interactions between B and T cells that may require the antigen to be first internalized by the BCR, expressed, processed, and then presented in an MHC restricted manner to T cells. B cells release a number of immunomodulatory cytokines that can influence a variety of T cell, dendritic cell, and antigen presenting cell functions, regulate lymphoid tissue organization and immunogenesis, regulate wound healing and transplanted tissue rejection. It also influences tumor differentiation and immunity. B cells can also function as polarized cytokine producing effector cells that influence T cell differentiation. Specific B10 regulates T cell mediated response by the production of IL-10. So B lymphocyte, natural AB, adaptive AB, auto AB, cytokine production, lymphoid tissue organogenesis, tumor immunology, transplant rejection, wound healing, dendritic cell regulation, Th1, Th2 cytokine balance, CD42, CD80, CD86, Ag presentation, B cell to T cell. Now let's have a summary. B cell subsets are a source of protective innate and adaptive antibodies. The phenotypic and functional diversity of B lymphocytes, their regulatory roles independent of antibody production, and their distinct molecular events makes this lineage unique. Mammalian B cell development encompasses a continuum of stages that begin in primary lymphoid organ, human fetal liver, adult bone marrow, with subsequent maturation in second lymphoid tissue. The functional endpoint is terminally differentiated plasma cell. B lymphocytes comprise 5% to 50% of total lymphocytic population. On encountering self-antigen, the development of immature B cell is arrested. On encountering foreign antigens, the proliferation and differentiation of B cell happen in the peripheral lymphoid organs. Isotype switching allows the variable regions of the antibodies which are responsible for antigen binding to interact with the constant region of any isotype, generating diversity in antibody production. Repeated antigenic exposure increases the population of memory cell which persist in the system for a long time. B cells secrete specific immunoglobulins in response to antigen stimulus, thus antibody-mediated immunity. Humoral immunity can be both active and passive types. 5 main classes of antibody are present in humans, namely IgA, B, E, D, and M. Apart from humoral immunity, B cells also mediate to regulate many other functions essential for immune homeostasis.